In this lecture, we will focus on how medicinal chemists and pharmaceutical researchers optimize the drug-target interaction to develop a lead compound into a drug candidate. Structure-activity relationships are at the heart of this practice. Once an initial lead has been identified, medicinal chemists aim to identify and optimize those parts of the molecule that are important to the biological function. In addition, they also identify regions of the molecule that have more flexibility and don't have a large impact on the biological function. These areas of the molecule may become focal points for optimizing pharmacokinetic parameters towards the later stages of drug development. The study of structure-activity relationships, or this idea that the chemical structure of a molecule determines its biological activity, dates back to the mid-1800s with investigations by Alexander Crum Brown and Thomas Fraser. In the example, you can see the structure of colchicine. This is not a pharmaceutical agent, but it's a compound that's used widely to study the biological process of mitosis or cell division. In the pharmacological context, colchicine disrupts microtubule polymerization and arrests cells in metaphase of mitosis, or the position of the cell cycle where the chromosomes line up at the middle of the cell and wait to divide into the daughter cells. In this molecule, nearly every functional group matters, and even minor structural variations can significantly alter the activity. Some key features include the 3,4,5 trimethoxyphenyl group, which is critical for activity. Removal of specific methoxys substantially reduce activity. In addition, the tropolone ring, or ring C, is also critical for the function of the molecule and cannot be expanded or altered without significantly reducing activity. So one of the first features that medicinal chemists consider when approaching structure-activity relationships is the pharmacophore. Pharmacophores are key molecular features that drive drug-target interactions. They represent the essential 3D arrangement of chemical groups responsible for a compound's biological activity. The concept is widely used in medicinal chemistry and computer-aided drug design to understand common structural and chemical features responsible for the biological activity of a set of compounds. While we're defining pharmacophores officially here, we've actually already been discussing these elements throughout the term and highlighted the use of pharmacophore features during the drug development of the potential hepatitis C treatment. And you can see that these features include all of the elements housed within the intermolecular forces that we reviewed way back in week 1. We have hydrogen bonding. We have hydrogen bond donors and acceptors, which can be visualized here in the antidepressive drug Prozac. Other features include hydrophobic regions or nonpolar areas of a molecule that tend to avoid interaction with water. These regions are often essential for the binding of the drug molecule to their targets, especially in the case of enzymes and receptors that contain hydrophobic binding pockets. Aromatic ring structures also exhibit unique electronic structural properties, and of course, any cationic, positively charged ions, anionic, negatively charged ions, and other dipole regions that carry partial charges can interact with receptor enzymes and stabilize interactions. Structure-activity relationship analysis using pharmacophore models identifies the essential three-dimensional arrangement of chemical features, like those hydrogen bond donors and acceptors and hydrophobics, for biological activity, helping to build rules that guide the design of more potent and selective drugs by correlating these features with experimental activity data. This can start with medicinal chemists synthesizing and testing a series of related compounds, which are called analogs, to see how changes in the structure, such as adding a methyl group or changing a ring structure, affect the potency, selectivity, or toxicity of the drug molecule. A pharmacophore model is built from these active compounds, pinpointing the recurring spatial pattern of essential features. Once a pharmacophore model is built, quantitative structure-activity relationships, or QSAR models, are developed. These are mathematically developed models that help link or identify pharmacophore features and sometimes steric or electrostatic fields to the measured biological activities, such as the IC50 or the EC50. The overall goal of the QSAR is to find the quantitative relationship between the structure and the observed activity of the drug molecule. This involves a combination of mathematical and statistical modeling. So what are these relationships good for once they're developed? QSAR models are built for several reasons. First, researchers want to understand the relationship between structure and activity so that they can design compounds with improved activity and predict the activities of novel compounds prior to their synthesis. This process often starts with a known training set of data. This usually includes compounds known to bind and inhibit your molecule of interest that were first identified as a hit from the library screening. From there, molecular descriptors are developed from the pharmacophore models. The program can then be used to search known chemical libraries or search for additional potential hits or leads. Ideally, these should include some known compounds that have biological activity towards the target that can then be used to validate the model and demonstrate that it's working correctly to identify lead compounds. Once the model is then validated, it can be used to screen in silico structures that have yet to be synthesized. So once a QSAR model is obtained and reproduces the known data satisfactorily, it can be exploited by computer algorithms, or AI, to predict the biological activity of not yet synthesized analogs. New predicted compounds can then be sent to the organic chemist to be synthesized, while non-active predictions are tossed aside. HIV drugs represent a big success story using this type of strategy. Scientists started with a simple dipeptide, phenylalanine and proline, that represent a rare cleavage site specific to HIV proteases, but not human proteases. By replacing the cleavable peptide bond with a stable hydroxyethylamine group, they created a pseudo-dipeptide that the enzyme could not cut. Through iterative structure-activity relationships, or SAR studies, researchers enlarged the molecule to better fit the protease's active site. At the amino terminus, the S3 region, the addition of a quinoline group improved binding and selectivity. At the C terminus, or S2 region, stability was improved by converting ester bonds to more stable amide bonds. In the S1 region, replacing the standard proline ring structure with a decahydroisoquinoline structure dramatically increased potency. Candidate selection with this process resulted in a compound named RO318959, which was later named saquinavir, which showed a 16,000-fold increase in activity over the initial lead compound. Other leads that became approved drugs include nelfinavir, lopinavir, and several others. Bioisosterism is a medicinal chemistry strategy where one atom or group in a molecule is replaced by another with similar physical chemical properties, size, shape, electronics, to create a new compound called a bioisostere that maintains or improves the original biological activity, leading to better drugs with enhanced potency, fewer side effects, or better absorption, metabolism, and distribution, or the pharmacokinetics. It's a fundamental tool for optimizing drug candidates by strategically tweaking structures to achieve desired therapeutic effects, like reducing toxicity or increasing stability. Here you can see several chemical examples of bioisosteres. For example, the conversion of a hydroxyl functional group to that of an amine or a thiol could produce a modified structure with similar biological properties. The second diagram shows that these modifications can be quite complex and varied for even a single functional group, the amide in this case. Thus, bioisosterism is a productive way to create small libraries of related compounds. An example wherein the 1,2,4-triazole was successfully installed as the amide bioisostere within a rigid ring system is found in the short-acting benzodiazepine alprazolam. Benzodiazepines are a class of drugs used as anxiolytics, sedative hypnotics, anticonvulsants, and muscle relaxants, and have achieved significant economic success. Diazepam was discovered in 1963 and was later approved for clinical use to treat anxiety, epilepsy, muscle spasms, and alcohol withdrawal symptoms. However, the half-life of diazepam and its metabolites are extremely long, greater than 24 hours, and this can lead to toxicity. Alprazolam was found to be more potent than the parent compound, did not produce active metabolites, and had a greatly reduced half-life in the body, significantly improving the toxicity profile of the treatment. And you can see the change is predominantly up in this region of the molecule. Conformational analysis represents another strategy for drug design. It's the study of the three-dimensional shapes or conformations that a molecule can adopt and how these arrangements influence its biological activity. Because most drugs are flexible molecules that rotate around single bonds, they exist as an ensemble of multiple shapes rather than a single static structure. The primary goal is to identify the bioactive conformation, the specific three-dimensional arrangement required for a drug to bind optimally to its target receptor or enzyme. Predicting affinity is an important component of conformer analysis. A molecule must often rearrange its shape in solution to fit into a binding pocket, and this rearrangement can carry energetic penalties that reduce binding affinity. Medicinal chemists can then choose or modify the compound further to induce conformational restriction. This is where the design of the drug is used to lock a molecule into its bioactive shape, such as by adding rings or bulky groups, which can improve potency and selectivity. Conformer analysis can also be used for pharmacophore modeling, where researchers use ensembles of conformers to identify common structural features among different active molecules, helping them to design new drugs. An excellent example of conformational restriction is an FDA-approved drug called cariprazine. This is used as an antipsychotic drug to treat schizophrenia. Cariprazine uses a rigid 1,4-disubstituted cyclohexyl ring to limit flexibility, locking the molecule into shape that fits better in the D2, D3 dopamine receptor targets, boosting potency and selectivity compared to the more flexible butyl chain analogs. Physicochemical properties like lipophilicity, solubility, the pKa, molecular weight are all crucial in drug design as they dictate a molecule's behavior in the body, influencing absorption, distribution, metabolism, and excretion, and the overall efficacy, safety, and drug likeness. Balancing these often conflicting properties, such as needing solubility for transport versus lipophilicity for membrane crossing, allows chemists to optimize potency, target engagement, permeability, stability, and to minimize toxicity, guide lead optimization, and predicting success. Towards finding an optimized strategy, Lipinski's Rule of Five was proposed in 1997 by Christopher Lipinski after analyzing the physicochemical properties of hundreds of orally active drugs. A compound is more likely to be orally active if it has no more than one violation of the following criteria. First, molecular weight should be less than 500 Daltons. Smaller molecules cross biological membranes more easily. 2, the log P should be less than 5. This is a measure of the lipophilicity. There needs to be a balance between solubility and membrane permeability. If it's too lipophilic, you'll end up with poor solubility and higher toxicology or toxicity risk. 3, there should be less than 5 hydrogen bond donors. These are typically hydroxyl groups and amine groups. If there are too many donors, molecules stick too strongly to water. 4, there should be less than 10 hydrogen bond acceptors. These are typically nitrogen and oxygen atoms with lone pairs. Excess acceptors reduce membrane permeability. Note that there are only 4 rules, but all the values cluster around the number 5 when optimized, hence the name, Lipinski's Rule of Five. The Rule of Five captures this solubility permeability trade-off in a simple empirical way. However, it's not a law and it's not universal. It does not apply well to larger natural products that have drug-like activities, such as the larger macrolide antibiotics that we looked at earlier. In these cases, active transport and other pro-drug strategies makes them successful in spite of violating these rules. You can see examples of these physiochemical properties in this diagram, showing lipophilic regions, the overall molecular weight, and the hydrogen bond acceptors and donors. Over the past decade, artificial intelligence, or AI, and machine learning, ML, have shifted drug discovery from a largely trial and error sequential process into a more data-driven, predictive, and parallelized workflow. AI does not replace chemists or biologists, but it augments human decision-making at nearly every stage of the pipeline. For example, identifying a biologically relevant target can take years with high failure rates. AI models can now integrate omics data, genomics, transcriptomics, proteomics, to identify disease-associated pathways and predict target disease relationships. AI can suggest novel or non-obvious targets. This is especially powerful for complex diseases such as cancer or neurodegeneration, which we'll talk about later this term. It can also help with polygenic disorders that involve multiple gene loci. AI excels at pattern recognition across massive, noisy biological data sets, something that humans struggle to do at scale. AI models are also being used to enhance in silico QSAR studies, conducting virtual screening and designing new drug leads. And one of the most valuable breakthroughs with AI has come in the form of transformative advances in the prediction of protein folding. AlphaFold is an example of an AI system that predicts a protein's three-dimensional structure from its amino acid sequence alone, a problem known as the protein folding problem. Why does protein folding matter? A protein's structure determines its function, binding interactions, stability, and drug targetability. Historically, determining protein structure required X-ray crystallography, NMR spectroscopy, or cryo-electron microscopy. These methods are slow, expensive, and not always feasible. AlphaFold stunned the field during a critical assessment of structure prediction competition, or a CASP competition. CASP is a blind competition. Structures are unknown at the prediction time. AlphaFold achieved nearly experimental accuracy. Errors made often fell within the margin of experimental uncertainty. This was widely described as solving the protein folding problem for many proteins. The image here shows the results from the CASP13 competition, where AlphaFold predicted structures that are shown in blue overlaid on the experimentally determined structures shown in green. So you can see just how well this model prediction worked. Overall, by using a combination of all the strategies presented, SAR is how medicinal chemists turn a hit into a drug candidate. We've explored how SAR investigates and optimizes chemical structure that controls biological response. These include functional group modifications, chain length and substituent alterations, ring substitutions, stereochemical modifications, the introduction of conformational restriction, and bioisosteric replacements. Medicinal chemistry seeks to improve drugs by substitution, not reinvention. It's extremely difficult to come up with a completely new chemical molecule that houses a desired biological activity. Chemists will usually start with an analog that has an existing biological activity and try to make improvements to that core structure. This is the process of creating the bioisomers, or creating molecules that have similar structure and similar function. The goals are to improve potency, reduce toxicity, and enhance stability. The bioactive conformation is the shape that fits the target. Oftentimes, small molecules are quite flexible and can adopt many conformations in solution. Chemists will try to introduce modifications that help lock the molecule into the bioactive conformation in an attempt to increase potency, improve selectivity, and reduce off-target binding. QSAR laid the groundwork for AI-driven drug design. Note that AI doesn't replace SAR, it scales it. SAR is no longer just about molecules, it's about molecules in the context of structural binding with their target molecule. This enables the possibility of predicting the activity of new or unsynthesized compounds. This can then significantly reduce experimental workloads by prioritizing potential lead compounds to investigate. Overall, modern drug discovery integrates SAR, QSAR, and AI to reduce failure, accelerate optimization, and make better drugs faster. And this is where we'll end our lecture on SAR.